So coming to the structure of proteins, so basically a protein can be considered to be a thread-like structure, a beaded thread obviously, because the beads represent amino acids which are held together by peptide bond. So you can consider a thread-like structure, a woolen thread-like structure, then this is referred to as the primary structure. So if a protein is just in the form of a linear chain, it is considered to be the primary structure. Always the first amino acid in this thread is considered to be the N-terminal amino acid, and the last amino acid in this thread is considered to be the C-terminal acid, because the first amino acid has a free NH2 group. There is no amino acid ahead of it to form a peptide bond, and the last amino acid has a free COOH group. There is no amino acid before it after it to form a peptide bond. What if this thread gets coiled into a spring-like structure like this, or if it gets folded around itself into parallel structures like this? Then we call it as a secondary structure. This is called alpha helix, where it folds itself into a helix, into a helical structure. It is a right-handed helix, and this is called a beta pleated sheath. A beta pleated sheath is where it forms a sheet-like arrangement. It may be more than one protein thread also, which are connected by bonds to form a sheet-like structure. So you have more than one protein thread which are interconnected by bonds to form a sheet-like structure, or the same protein thread which is folded upon itself in order to form a beta pleated sheath. Or you take a thread and you coil it like a spring, then that becomes the alpha helical structure, and this pleated form, both of them represents the secondary structure of proteins. What if the protein chain looks like a woolen ball? You've taken a thread and you've rolled it into a woolen ball, then it is called a tertiary structure. And it is very, very important to note that the tertiary structure actually represents the three-dimensional structure of a protein, and it is very, very important for a protein to exist in its tertiary structure in order to be able to perform its biological activity. So it's a functionally active form, active structure. What if you have more than one woolen ball with you? It's like more than one polypeptide chain, more than one protein. Each individual protein is in its tertiary structure. Then the entire architecture is referred to as a quaternary structure. One woolen ball is a tertiary structure. Say you have 4 woolen balls in a box. That represents a quaternary structure. The best example for quaternary structure is hemoglobin molecule. Hemoglobin molecule has 2 alpha and 2 beta protein chains, 2 alpha and 2 beta in a part of it called globin protein part. So this actually represents a 4-degree or a quaternary structure, because each circular structure I've shown there is a tertiary structure. So it's basically 1, 2, 3, 4. Each of them are in their tertiary, 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 tertiary. The whole thing becomes a quaternary structure. So it's basically like a bunch of woolen balls with you. So when you have a bunch of woolen balls, it makes up a quaternary structure.